So today we will learn the chapter 1. The chapter 1 title is Overview about Epigenetics. Chapter 1 is composed of the 8 subjects, start from the centromere and epigenetics, and also there are several epigenetics models, and finally we will learn about pervasive transcription. Let's start from the first subject, centromere. So what is centromere? So this is the centromere, and this is the centromere. So centromere is the specialized DNA sequence of the chromosome that links a pair of sister chromatid. So this is they linked each other, so it will make the chromosome. So centromere is centromere is a specialized DNA sequence of chromosome that links a pairs of sister chromatid. So next to the centromere, there is a kinetochore. So this is centromere. Next to the centromere, there is kinetochore. Kinetochore is composed of the inner kinetochore and out kinetochore. So usually the kinetochore is a complex of proteins associated with the centromere. And also especially the kinetochore, especially the out kinetochore is place where spindle fiber, this is spindle fiber, in Korean we call it as the 방추사. Spindle fibers attaches during the cell division. Okay. So in this slide we will learn about several kind of the centromere. Start from the point centromere and hollow centromere and regional centromere and statewide centromere. Okay, let's start from the point centromere. So point centromere in Korean we call it as 점 중심절. Okay, 점 means that the size is very small. Okay, so point centromere is a specialized DNA sequence of chromosome that links pairs of sister chromatid. But in 점 중심절 point centromere, the size is about 125 base pair, only 125 base pair. So this point centromere is only observed in the Saccharomyces cerevisiae. So this is the birding yeast. They always reproduce by birding, and their birding yeast is very lower classes of the eukaryote. After birding yeast, that there is a C. elegans, and after more developed organism is the fission yeast. After that, the mammalian, something like that, like this. Okay, so this is only observed in the yeast, birding yeast. And also, okay, how about in this region, the point centromere is composed of CDE1 and CDE2 and CDE3. So it means that in this region, CDE2 is the specialized the DNA sequence, chromosome centromere sequence where the CEMPA is bind. Okay, so this is the chromosome. Each chromosome is composed, the basing unit is nucleosome. So nucleosome is composed of the histone proteins, 8 histone proteins. Start from the 2 H3 histone and 2 H4 histone and 2 H2B and 2 H2A histone. So usually this is histone protein. After that, just the DNA is wrapped. The histone proteins is like this. But in the centromere region, so this is the DNA sequence. But in the histone protein is different. For example, in the chromosome region, arm region, so there is a H3 histone. But in centromere region, there is a CENPA histone proteins. They have two CENPA histone and two H4, two H2B, and two H2A proteins, histone proteins. So usually in this site, usually the CENPA histone protein is bind to this region. Okay. Okay, how about the holocentromere? So holocentromere in Korean we call it as a 유사중심절. So this 유사중심절 only observed in C. elegans. So holocentromere is spread over different parts of the chromosome. So each holocentromere, its size is about 17 megabase. But usually we already see that centromere is located in the center region of the chromosome. But the holocentromere means that this centromere is spread all over the chromosomes like this. So this phenomenon is only observed in C. elegans. How about the regional centromere? So regional centromere in Korean we call it as the 국부중심절. So this is the most popular form of centromere. It is observed in the fission, start from the fission east, and also in mammalian, all of mammalian, they have the regional centromeres like this. The size is about 4 or 7 kb. Okay, for example, and also they have the regional centromere is usually observed in the fission east. So they reproduce by fission. Okay, for example, this one is they reproduce by budding, but this is they reproduce by fission. So that is piece each other like this. So and okay, so usually the regional centromere is observed in the fission east and also observed in all of the mammalians. Okay, regional centromere is composed of the pericentric heterochromatin region, and also inside there is the CENPA binding region. This is the CENPA binding region. Okay, but the DNA sequence in the regional centromere is not conserved. Why? Because this is near centromeres. So we will see this concept in the next slide. Okay. Okay, and also there is another centromere. We call it as a statewide centromere, 부수 중심절. This is only observed in the mammalians. So this is the tandemly repeating and non-coding DNAs. For example, this is the only observed in the mammalian. So the size is about 1 to 2.5 megabase. So they also have the heterochromatin region, pericentric heterochromatin region, or inside they have the tandemly repeat. One is the CENPA or binding region. The other is not the CENPA binding region. And next there is CENPA binding region, and this is tandemly repeating centromere. A little bit different from the regional centromere. In regional centromere, in the inside region, and this region is in all of this region, CENPA can bind to that region. But in the statewide centromere, is only this CENPA only bind to the tandemly repeating region. So this is, we call it as the statewide centromere. Okay, now we will learn about neocentromere. Okay, we mentioned that the regional centromere is usually the neocentromere, and means that the DNA sequence in the original centromere is not conserved. Why? So we will see the reason. Okay, so this is the chromosome, is like this. So chromosome is composed of the short arm. They have the short arm, and they also have a long arm. They have a short arm, long arm is like this, and this is the AC. So AC region is the we call it as ancestral chromosome. So this is the chromosome ancestral centromere. So this is the centromere region. So original centromere is located here. Okay. After that, the gene A, gene B, gene C, gene D is located in the short arm, and gene E, gene F, and gene O is located in the longer arm. So sometimes, usually the chromosome deletion happen. So in this case, is the centromere is the disappears. So original centromere is disappears. So original centromere is usually the centromere is a little bit wider, but the original centromere is disappears. So they only have a short centromere is like this, but in this case is the new centromere is the forms on a chromosome at a location that is normally not centromeric. So this is the usually it was here between the centromere is located between gene D and gene E, but in this neocentromere is the location is not normally in the centromeric. So usually the neocentromere occurs on the most chromosome, but mostly found in the longer arms of chromosome 3 and 4, 8, 13, and 15. They usually seen in the longer arm of the chromosome 3, 4, 8, 13, 15. But this is also occurs in almost most chromosome. So that's why the DNA sequence in the neocentromere is not conserved. So usually in this region, the centromere region DNA sequence is very conserved, but in the newly formed neocentromere region, the DNA sequence is not conserved. So that's why we call it as this is neocentromere. So this neocentromere is only observed in the original centromere. Okay. So in this slide, we will learn about some terminology, synteny. So synteny means that, for example, this is the comparative analysis of human genome. This is human genome and the macaque 원숭이, macaque genome. So this is macaque genome. They are almost 90% identical in genome, and also even in gene composition and gene order. So synteny means that physical co-localization of genetic loci on the same chromosome within species. For example, this comes from human, and comes from the macaque. Okay, so gene location is there. Gene A is located here in human, and gene A is located in here in the macaque, and gene B is located here in human, and gene B is located here in macaque, and gene something like this. So synteny means that physical co-localization of genetic loci on the same chromosome within species. Okay, but in the synteny in centromere does not exist. For example, this is one example, but this is the usually the normal chromosome structure, and the centromere is located in between the gene D and gene E. For example, this is human. How about macaque? Macaques, this is macaque. So usually the physical co-localization of gene is the gene order is nearly similar to the human. Start from the A, B, C, D, something like that. But the centromere location is around here. So for example, centromere location is different on same chromosome within species. For example, this is the human and this is macaque. So this means that synteny in centromere does not exist. So centromere is not physically co-localized. Okay, so in this slide and previous slide and previous slide, so we learned about the centromere. Why we learned about centromere in epigenetics? So we will cover this subject in the chapter 2 and chapter 3. Why we cover the centromere in epigenetics? So usually the centromere is we can see the highly epigenetic phenomenon in the centromere. That's why first we start from the centromere in subject 1. Okay, and so we will learn about the centromere details in chapter 2 and chapter 3. This slide we will explain the second subject, definition of epigenetics. So we will define what is the epigenetics. So in last semester, you guys already learned about genetics. It means that characteristics of father is usually the transferred to the offspring children. For example, if the father is taller, maybe their children have a lot of tendency to their height is maybe taller. Children is also very taller. So it means that some characteristics is inherited from father to children. But the epigenetics, so epigenetics means that epi plus genetics. Epi means that after or beyond genetics. So epigenetics means that gene function is also can be inherited from father to offspring. Some characteristics can be inherited from father to offspring, but without changing the DNA sequence. So in genetics, usually some changes in DNA sequence is changed to their offsprings. So, for example, if their father has the longer height, it means that their father has some gene, some specific DNA sequence which encode the longer height, and also their offspring is inherited that DNA sequence. But in epigenetics means that epigenetics is a study of changes in gene function that can be inherited without changing the DNA sequence. So usually the epigenetics is DNA methylation and a lot of histone modification. And also there is a non-coding RNA is also very involved in very important role in epigenetics. So we will learn about chapter 4 or chapter 5 and DNA methylation. Also we will learn about histone modification in chapter 6 or chapter 7. And also we will learn about non-coding RNA in the later chapter. So this is epigenetics. Important thing is a study of changes in gene function that can be inherited without changing the DNA sequence. Only changes in the DNA is some DNA base is methylated or histone protein is modified, and also some non-coding RNA is involved in epigenetics. So in this slide, we will learn about another definition of epigenetics, which call it as epigenetic landscape. Okay, so during normal development, so a lot of the epigenetic things happen. So DNA methylation and histone modification is happen. So after that, pre-differentiated cell is due to several epigenetic changes. This pre-differentiated cell is changed to the differentiated cells. But in the epigenetic landscape, usually the differentiated cell unable to return to their original pre-differentiated cell. So this is the epigenetic landscape. But nowadays, this epigenetic landscape concept is changes. So we call it as modified epigenetic landscape. So there is the pluripotent reprogramming, and also there is the direct conversion. So during the pluripotent reprogramming, so differentiated cell is changed to the pre-differentiated cell. After that, this pre-differentiated cell can be changed to differentiated cell. It's due to changes of several DNA methylation or histone modification or non-coding RNA. Due to the non-coding RNA, such cells can be changed to the pluripotent reprogramming happens. So after that, differentiated cell changes to the pre-differentiated. Finally, it will change it to differentiated cell. Or there is also another possibility is direct conversion. It means that differentiated cell can change it to the differentiated cells. It means that skin cell can change it to liver cell, or skin cell can change it to the hair cells. This is, we call it as direct conversion. It's due to several epigenetic changes. Okay. So now is this is a traditional concept of epigenetics. Nowadays, epigenetic concept is changes. This is modified epigenetic landscape. So in this slide, I will explain the third subject is that is epigenetic information inherited? What is your answer? Is epigenetic information inherited? Is yes. So I will show you some example. The very famous example is some specific gene. The gene is the Algorti locus. Okay, so gene name is Algorti. So okay, this gene is regulate the production of pheomelanin. So usually the pheomelanin make the yellow color, and also it can regulate eumelanin. So eumelanin color is the black. So usually this Algorti gene locus regulate the production of the pheomelanin and the eumelanin. So in this locus determines hair color in mice. Okay, how about for example, there is the allele large A or allele small a's. For example, large A is dominant than small a. Okay, so if there is a large A, it means that pheomelanin is produced more than eumelanin. So if there is allele large A is located, so pheomelanin is the highly produced, and eumelanin is the produced is very small. Most of them is pheomelanin. That's why the hair color will be the yellow. So large A, the mice which has large A, their hair color will be yellow, and also large A, small a is also their hair color is yellow because large A is dominant than small a. But allele small a is mutant form of allele large A. So in these cases, they cannot produce pure melanin, so they only can produce the eumelanin. That's why it can be the black. So the mice which has the genotype small a, they have the black color. Okay. So in this slide, I will explain how the agouti locus is epigenetically affected. So, okay. Now is some scientists inserted intracisternal A particle into the promoter region of the agouti allele, large A allele. So initially, these mice, the genotype is large A, small a, but now the genotype is changed to large A, VY, and small a. So usually in the IAP region, there are lots of the CPG island. So CPG, usually the DNA methylation is happen in the CPG island. Okay, so usually the DNA methylation on CPG island control the TSS. TSS means that transcription start size. So there are two cases here. So if this region IAP region is hypo, there is no methylation. Usually in these cases, it can activate TSS. So at that the agouti large A allele is highly activated, expressed. In these cases, the pure melanin is highly produced. At that the color, the coat color will be the yellow, is like this. But sometimes this region is hypermethylated. So in these cases, when they are hypermethylated, there are two cases happen. So sometimes it can inactivate the expression of A allele, so the transcription start site is inactivated. So there is no expression of large A allele, but sometimes it can activate the large A allele during some specific point. So their final color is the yellow. When they inactivate the large A allele, they will make the black color. So when they are sometimes they activated, they will make the yellow color. So finally, their color will be they mixed the yellow and the black. Finally their color will be the browns like this. So this is brown color. Okay. So now we will learn about epi allele. So epi allele is the genetic locus which is affected by DNA methylation. So large A allele is also epi allele. Also, we will also learn about another allele, DNMT3. Okay, it is later. Okay, so this is the large A VY is the epi allele is affected by DNA methylation. So okay, is in these cases that so they use the agouti large A VY and small a mouse model. So for example, in one case is if we treat the same mice, so if we treat the isoflavone, it comes from the bean. So they extracted the isoflavone. In this case, when the isoflavone is treated to mice, it can induce methylation in the own IAP region. So in this case, it can inactivate the allele A and sometimes the activated expression of large allele A. So finally, their coat color will be brown. It's like this. But usually, when the coat color is brown, usually these mice is very thin and very healthy. But when sometimes the BPA bisphenol A is treated to these mice, the same mice. So in BPA is do not induce methylation in the IAP. So in this case, it can activate the large A allele. So coat color is yellow. But in this case, when the large A allele is activated, they have also another function. The mice will become very obese. So in this case, these mice is prone to cancer and diabetes. So the mice which has coat color is yellow, they are very obese and they are also prone to cancer and diabetes. When the coat color is the brown, they are very thin and healthy. So this is the one example of epi allele. So another example is we can see in the bees. So okay, so actually the genotype of queen bee and the work bee is same. They are start from the same thing, but in one group we treated royal jelly, but the other group we treated just the work jelly. So when royal jelly is treated, so this same larva is developed to the queen bee, but the work jelly is treated to the same larva. So this bee will be the work belly. So this is also epigenetic findings. So for example, royal jelly is inactivated DNMT3 gene. So DNMT3 is the DNA methyltransferase. So it means that in inactivated DNA methyltransferation, so it means that there is no methylation. So it means that there is no methylation in the DNA. It means that it can induce queen bee. But when the work jelly is treated to the same larva, so usually this work jelly is activated DNMT3. So it means that there is a loss of methylation. So it will make the work bees like this. Okay. So the important thing is that is epigenetic information is inherited? Yes. So inherited. These mice, the offspring will be the thin and healthy mice, and these mice, the offspring will be the obese mice. Okay. So from this slide, we will learn about fourth subject, component of epigenetics. Okay. So now we will start from the DNA methylation. After that, we will also cover the histone modification. Okay. Let's start from the DNA methylation. So what is DNA methylation? Usually the cytosine, the base cytosine, this is cytosine. So usually in the 1, 2, 3, 4, in fifth location, this carbon is usually the fifth location of the carbon is methylated. So usually the methylation happen in the cytosine, especially in the fifth location is carbon. So we call it as a 5-methyl cytosine. So usually the methylation is due to by DNA methyltransferase. So this is the DNMT. So DNA methyltransferase, DNMT. So usually it can induce DNA methylation, especially at the cytosine. So there are three kinds of the DNA methyltransferase. First one is DNMT1, and the other is DNMT3A and 3B. So they have a different function. So we will cover some detailed information of DNA methylation in chapter 5. Okay, chapter 5. Okay, now we can go to some of the specific function of DNMT1 and DNMT3A or DNMT3B. So DNMT1 and DNMT3A, 3B, they can do the DNA material transferase function, but they have some different function. Okay, the first one is the DNMT1 is a recreate methylation pattern on hemimethylated DNA. For example, so this is the DNA, double-stranded DNA. So usually one strand is the methylated. So methylated DNA is the replicated. So in this case, the one strand is methylated, but the other newly synthesized strand there is no methylation is here. But in this case, the DNMT1 recreates the methylation pattern on the only one strand is methylated. This is hemimethylated DNA, so they can recreate the methylation pattern on hemimethylated DNA. But how about DNMT3A and 3B? So this can do the de novo DNA methylation. For example, this is DNA double strand, there is no methylation. But de novo methylation means that it can induce DNA methylation in the newly synthesized DNA methylation. So it can induce the methylation in both sides is like this. This is we call it as a de novo methylation. It is caused by DNMT3A and or DNMT3B. So this is the main difference between DNMT1 and DNMT3A and 3B. So we will cover some detailed information in chapter 5. So now we will also learn about another component of the epigenetics, which is the histone. Okay, so we already learned that histone octamer. Okay, so we already learned that so histone is the histone octamer in nucleosome. So this is nucleosome is composed of the 8 protein complex at the center of a nucleosome core particle. Is composed of the 2 H3 histone protein and 2 H4 histone protein. So usually the H3 and H4 they make the dimer, and also finally they will make the tetramer is like this. So usually the H3 and H4 usually they made the dimer is like this. So finally they will make the tetramer, H3 H4 tetramer. So in this case, at that also there is another 2 component which is H2B, 2 H2B and 2 H2A. So usually the H2A, H2B is they make the dimer, and also H2A, H2B they make dimer. But in this case is in this region the H2A, H2B bind to this region, and also another dimer is bind to this region. So finally they will make the histone octamer is like this. Okay, and this is histone octamer. And what is nucleosome? Nucleosome is composed of histone octamer and the DNA. This is a left-handed superhelical turn. So DNA is composed of the 147 base pairs of DNA. So it means that one turn and 0.7 turn. So it can induce 1.7 left-handed superhelical turns. So we call it as nucleosome. Okay. Now we can go to the next slide. Now I will explain another component of epigenetics, histone modification. So we already learned about histone, the histone complex, especially the histone octamer, and also we learned about nucleosome. But usually in histone there are lots of the modification happens. For example, so this is a histone. So histone have their own tail. For example, this is the H3 protein, H3 histone protein, and this is the H4 histone protein, like this. For example, usually the histone modification happen in the histone tail. So histone tail, there are lots of the acetylation and methylation, phosphorylation, and ubiquitination happens. So this is histone H3 and H4. This is H2A. This is H2B. It's like this. So usually all of the chemical, nearly 99% chemical modification happen in the tail. But sometimes just less than 1% also chemical modification happen in the inside of the hydrophobic region of the histone. Sometimes happens, but not much, just less than 1%. So most of the histone modification happen in the histone tail, especially the acetylation and methylation and phosphorylation and ubiquitination. All of these changes is epigenetic changes. So these changes can be inherited to the offsprings, your offsprings. So in this slide, I will explain the histone code hypothesis. So there was a lot of histone modification here. So in this slide, how to decode the histone code. Okay. So usually the transcription of genetic information is partly regulated by chemical modification in histone tail. So there should be some material who can decode the histone code. So the material that decodes the information of the histone modification exists. So material can be protein or RNA. So usually this is bromodomain. So the bromodomain, so this is proteins. So it can, this bromodomain is recognized the histone acetylation. So bromodomain decodes the information of histone acetylation. So sometimes it can activate the transcription or inactivate the transcription. For example, and also there is other domain existed. For example, if there is some methylation here, another domain also can decode histone methylation code. After that, so it can activate the gene. It can activate transcription, or it can inactivate transcription. So usually the nucleotide sequence can be the genetic information, and nucleosome, because the nucleosome contains the histone octamer, so it can have the epigenetic information. So histone code hypothesis, once again, the transcription of genetic information is partly regulated by chemical modification in histone tail. So usually the bromodomain decode the histone acetylation. Usually the bromodomain recognizes the histone acetylation and decodes the information. And also there is other domain exists. Sometimes some domain is recognized methylation, and some domain is recognized ubiquitination and something else. So nucleotide sequence can be the genetic information, and nucleosome can be the epigenetic information. So in this slide, I will explain the fifth subject, is a position effect variegation. So position effect variegation is the molecular mechanism of the epigenetics. So what is position effect variations? PEV is the usually sometimes in the gene, in the chromosome or in the gene arrangement is always happen. So sometimes gene arrangement induces abnormal juxtaposition of gene with a heterochromatin. So today I didn't explain what is heterochromatin. So now I will explain very briefly about heterochromatin. So usually the heterochromatin is very condensed, the region. Usually this region is observed in the telomere region or in the centromere region. But what is the opposite? Opposite is euchromatin region. The condensation is very relaxed, something like this. Okay, so usually in this case is the ADE2 gene is located far away from the heterochromatin region. But some gene arrangement happens. So gene arrangement induces abnormal juxtaposition of the gene, especially ADE gene, with heterochromatin region. So in this case, actually the ADE2 gene was located in the euchromatin region. Euchromatin region means that very relaxed region. But now is ADE2 gene is located in the heterochromatin region. It means that very condensed region. So actually the function of ADE2 gene is to produce the white cell, white colonies in the yeast. So this is the ADE2 positive cell. The yeast they show that the white colonies. So in this case the ADE2 gene is far away from the telomere. So it means that they are located in euchromatin region. So it means that the heterochromatin region cannot inhibit the expression of ADE2 genes. It means that it can produce the white cell. How about the gene arrangement happens? So ADE2 gene is the abnormally juxtapositioned with the heterochromatin gene. So in these cases, so ADE gene is located in the gene is put in environment with a different condensation. In this case, they were located in the very condensed region. So in these cases, near to telomere, so it inhibits the heterochromatin region, because heterochromatin region is very condensed. It means that it inhibits the expression of ADE2 gene. So it means that it can produce lead colony. Initially, ADE2 gene have to produce white cell, but in this case, ADE2 gene is repressed, sometimes repressed or sometimes activated, so it can make the lead cell, and also with the white sector is like this. So this is the ADE2 negative genes. Okay. So in these cases, the ADE2 gene is juxtaposition happens. So gene arrangement happens. So ADE2 gene is near the heterochromatin region. Okay, this is the position effect variegation. Okay. So in this slide, I will explain another PEV example. So we can see this phenomenon in white allele in the Drosophila. So in Drosophila, there is a white gene is located in the Drosophila. White gene is there. So for example, this gene, there are two alleles. One is large W allele, the other is small W allele. And large W is dominant than small w. Large W allele encodes the red pigment, and small W allele encodes white pigment. Because the large W is dominant than small w, in this case large W, they will show the red eye. How about another case? Large W, small w, they will also show that red eyes. How about in the case of small w? In this case they will show the white pigment, white eyes like this. So when the white allele, white gene is away from the chromatin, so white gene is away from the heterochromatin. For example, this is heterochromatin. White gene is located here. So this region is very condensed and this region is very relaxed. So when the white gene is away from the heterochromatin, so in this case, heterochromatin cannot inhibit the expression of allele large W. So in this case, the phenotype will be the red eyes. So when the white gene is near to heterochromatin, so in this case some abnormal gene arrangement happens. So in this case the white gene is very close to the heterochromatin region. So in this case white gene is very close to the heterochromatin region. For example, this white gene is inside of the heterochromatin region. This region is very condensed. When the gene is located in the condensed region, the expression of the white gene cannot be expressed. So usually this situation inhibit the expression of allele large W. So in these cases, the phenotype is the red eyes with white spectra, It means that sometimes this white gene large allele large W can be expressed, but sometimes cannot be expressed. So the phenotype is red eye with white sectors like this. So this is another example of the PEB. So in this slide, I will explain the molecular mechanism to explain the PEB. So as I mentioned earlier, so euchromatin region can be changed to the heterochromatin region. For example, it was the gene A is located in the euchromatin region, but sometimes the gene arrangement happens, so this gene can be located into the heterochromatin region. Or sometimes when gene is located in the heterochromatin region, gene arrangement happen, so they can be changed to the region. The location can be changed to the euchromatin region. So it means that they are interchangeable like this. So usually the gene near the heterochromatin region, so within the heterochromatin region, so it means that the several transcription factor cannot bind to this heterochromatin region. So it means that it can induce inactive transcription of gene A. But when the gene A is away from the heterochromatin region, so it means that in this region is euchromatin region. So usually the several transcription factor can easily bind to this region. At that, it can induce active transcription of gene A. So this is the underlying molecular mechanism of PEB. Okay. So in this slide, I will also explain some basic principle of the position effect variations. So we have to understand the flexibility of heterochromatin formation. So usually this phenomenon is happen during S phase. So sometimes, for example, sometimes the gene is located, one gene is located in the heterochromatin region. Okay, so usually during the S phase, DNA synthesis happens. So during DNA synthesis happens, usually the heterochromatin region is relaxed. So it can be euchromatin. After that, the DNA synthesis happens. So one strand can become to the two strands is like this. So during this time, is the heterochromatin region is the flexibly interchanged to the euchromatin region. And also during S phase, mRNA synthesis is also happen simultaneously. So the following process is very difficult to understand. So I will just briefly explain the procedure. Okay. So usually, during the DNA synthesis is happen, the mRNA is also synthesized simultaneously. So it usually the recruit RNA polymerase. So after that, the mRNA is transcribed. After that, this mRNA is usually the recruit dicer complex. So dice complex is generated. They chop the RNA. So they will make the small siRNA, very small RNA. After that, this siRNA and R1 protein they make the complex. And this complex is go to the moved where the mRNA synthesis happens. So usually there is a H3K9 methyltransferase. We call it as the CLR4. So usually the CLR4 is methylated the DNA. And after that, it also recruits SWI6 protein. So after that, this region is become the heterochromatin regions. Okay, there are if in the late chapter we will see some details. Okay, but the important thing is that heterochromatin formation is very flexible. Okay, something like this. Just the heterochromatin is changed to euchromatin, and euchromatin is changed to heterochromatin, and this phenomenon is usually happen during the S phase, because during S phase DNA synthesis is happen. So heterochromatin region can be released and changed to the euchromatin region. After that, the DNA synthesis is happen, and simultaneously an mRNA synthesis is happen, and there are several step processes done. After finally CLR4 they did the methylation, and also they recruit SWI6 protein. After that, this region is become the very condensed heterochromatin region. So if we want to know some detailed information, I will teach you in the chapter 9 or chapter 10. Okay. And the very important thing is that PEB happens. Sometimes the gene arrangement happens, so it can induce the PEB phenotype. So they will show this kind of phenotype, or they will show this kind of phenotype. So the basic principal molecular mechanism to PEB is the euchromatin and heterochromatin. They are interchangeable. So okay, sometimes euchromatin is changed heterochromatin, heterochromatin changes euchromatin. And also we learned about this phenomenon is usually happen during S phase when DNA synthesis happen and mRNA synthesis happen. So heterochromatin changes to euchromatin. After euchromatin is changed to heterochromatin. It's like this. Okay. So from this slide, I will explain the sixth subject. It's a research method in epigenetics. So usually in epigenetics, we usually use the chromatin IP. We call it as a chip. So by using the chromatin IP, we can analyze which region, which genomic region is methylated or acetylated. Finally, we usually do use the big data analysis. Okay, so I will briefly explain some details in the chip and the chip-seq analysis. Okay, first one is the chip, chromatin immunoprecipitation. So usually for chromatin immunoprecipitation, first one is just retreated formaldehyde. So it means that protein, the histone protein and DNA binding is fixed like this. So usually without formaldehyde treatment, and usually the protein and DNA is dissociated. But after formaldehyde treatment, the DNA is permanently stick to the protein. So after that, we can do the fix the protein and DNA bindings. It's okay. So in this region, this is the heterochromatin region, and this is euchromatin region. So usually in the euchromatin region is usually the highly acetylated. Okay. So okay, this is one example. So usually we do the mechanical pulse, or we can sometimes do the enzyme treatment. After that, we can cleave the chromatin. Okay. So after we treated the mechanical pulse, heterochromatin region is also can be cleaved, and also euchromatin region also can be cleaved. Okay. Before explain, so this is heterochromatin region. So there is no acetylation can be found, but only in euchromatin region we see the histone acetylation. So this is histone acetylated nucleosome. Okay. After that, we just use the precipitation. So in these cases, we use the antibody against acetylated histone. So it means that this antibody is only precipitated acetylated histone. So it can only precipitate the acetylated histone. After that, we just only purify the DNA. After that, we can just signal amplify, or we can use the signal amplification using some specific PCR primers. So this is the chromatin IP. Okay. So based on this analysis, we will see if some gene is located in the euchromatin region. So if we use the gene primer, so we can amplify the gene here. So if some gene is located in the heterochromatin region, so after we use that gene primer, we cannot amplify the signal. So this is chromatin immunoprecipitation. So nowadays people also combine the sequencing technology in the chip. Okay. So chip-seq is a method used to analyze protein interactions with DNA. Okay. First criteria, people usually do chromatin IP. So okay, in these cases, For example, this antibody can recognize acetylated DNA, or sometimes this antibody, we can use another antibody which can use the methylated, the antibody can recognize methylated histone. Okay, after that just precipitated. After that we can release the DNA. After that we can do the sequencing. So we can do the sequencing. Okay, so finally we will know that which region. So if we use the antibody against the methylated histone, so we will know that which region is highly methylated and which region is not methylated. So this is the ChIP-seq. So to make this kind of graph, we have to, so the ChIP-seq data is analyzed by using big data analysis technology. So this is the raw data. This is the raw data read. So some region is red color and some region is the blue color. This is sequence. This is raw data. After that, so just they aligned this region with just genome. After that, we can make the by using the simple coding. This peak is obtained by processing raw data read. After that, we can make this kind of peak is like this. So some genome region is methylated, or some this region is methylated like this. So usually in the ChIP-seq analysis, after we did the sequencing, after we finish the sequencing, so to make this kind of peak, we always do the big data analysis. But today we will not cover in detail in big data analysis. So in this slide, I will explain the seventh subject, the regulation of gene expression by epigenetics. But to understand this subject, we have to know the promoter structure. So this is the promoter structure of eukaryotic cells. So eukaryotic cell in the promoter, they have the Tata box, and next to the Tata box, they have the TSS site, transcription start site. Next to the TSS site, there is a downstream promoter element, DPE. So Tata box is your consensus sequence characterized by repeating TA and A base pairs. So usually Tata box is the TAA. So this is the binding site of the Tata box binding proteins, TBP. So this is a Tata box region. So usually the TBP is bind to the here. Okay. Usually the 80% of promoter in eukaryotic cells encoding proteins do not have a Tata box, but we don't know why. Only 20% of promoter in eukaryotic genes encoding protein do have the Tata box. Okay. Next to the Tata box, there is a transcription start site where here around the start, this is composed of YYCA, YYYYY. So Y is pyrimidine. Pyrimidine is cytosine or thymine. So cytosine or thymine is go to the here. So YYCA. So usually around the A site, the transcription is started. So after that, there is the DPE region is located next to the TSS. So usually this is the binding site of TF, transcription factor 2D. So this is the transcription factor 2D. So after Tata box is bind, it recruit Tata box transcription factor 2D. After that, they also recruited A, F, B, or something else, finally they will recruit RNA polymerase 2. So this is the pre-initiation complex. Okay, so we understand the promoter structure. So next I will cover NFR. So what is NFR? So NFR is nucleosome free regions. So where is this is located? Usually located in the promoter region. And so in promoter region, there is no nucleosome. So in promoter region, usually in promoter region, there is no nucleosome. So this is recorded as NFR. So usually in NFR region is just nearly like the promoter region. This is the place where transcription factor can bind. So who make this NFR region? So who make this NFR region? So usually the HAT, histone acetyl transferase. So histone acetyl transferase to make this is the heterochromatin, condensed chromatin. So usually the HAT is transferred to acetylation, induced acetylation. After that, this chromatin is very relaxed, is like this. Okay, so usually they make the NFR region here and here and there, or ATP dependent chromatin remodeler and SWI-SNF complex. So this is SWI-SNF complex. This complex is also remodel the chromatin structure. After that, it can make the NFR region. Okay, so usually the HAT or SWI-SNF complex makes NFR region. So NFR region is the place where the transcription factor can bind. Okay. Next, I will show you. I will explain the transcription initiation in eukaryote cell. So usually the HAT or SWI and SF complex, it changes the chromatin structure. After that, it will make the NFR. Finally, so it will recruit several transcription factor. Finally, they will make the free initiation complex. So this is promote region. Finally, RNA polymerase bind to this region. So we call it as this is a pre-initiated complex. After that, RNA polymerase itself has the HAT function. So this RNA polymerase 2 itself also has the HAT function. So it means that it also induced histone acetylation. So it means that it will induce nucleosome eviction. So it means that it can evict the nucleosome. So it can RNA polymerase 2 can bind to the DNA region. After that, the transcription is start around here, like this. So transcription is start, and mRNA can be made like this. Okay. How about start from the once again? So this is a transcription initiation in eukaryote cell. Okay. Usually the HAT or SWI-SNF complex makes the space, makes the NFR region. After that, it can induce changes in chromatin structure. Finally, it will make the NFR, so it will recruit several transcription factor. So it will make the pre-initiation complex. And also RNA polymerase 2 itself also has the HAT function. So RNA polymerase is bind to the they make the pre-initiation complex at NFR region. So after that, they will meet the nucleosome. Okay, so this is the NFR region. Finally, they will meet nucleosome. So in this case, because the polymerase 2 has the HAT function. So it means that it can induce nucleosome eviction. So it can easily bind to the DNA. So finally, it can induce transcription initiation. So this is the regulation of gene expression by epigenetics, especially by HAT, OR3, and SNF complex. Okay. So in this slide, I will explain the final subject, pervasive transcription. So what is pervasive transcription? So in our genome, the genes that we are interested in are only 2% of genome. So it means that we can expect that only 2% of gene is transcribed. But actually we found that transcriptions occurs in 93% of the genome. So this is the pervasive transcription. Why this kind of phenomenon is happening? Okay, for example, if we are interested in this gene is interested in the gene. So usually the transcription factor is binding to here. So only this gene can be transcribed. Okay, for example, this is gene 1 and gene 2. So only gene 1 is transcribed by the transcription factor, and also only gene 2 can be transcribed from the transcription factor. But we found that the transcription occurs in the 93% of genome. Transcription occurs nearly in everywhere. So due to the pervasive transcription, so hundreds of transcript can be transcribed from the one gene. So actually, usually the one mRNA should be transcribed, but we found that hundreds of transcripts can be transcribed from the one gene. Why this kind of phenomenon is happening? Okay. In this slide, I will explain the underlying mechanism to promote pervasive transcription. So usually the chromatin-associated protein, HAT or SWI and SNF or histone chaperone. So this protein is induced histone deassembly or assembly. So doing the transcription elongation. So if we remove this gene, these proteins, sometimes it always induced pervasive transcription. So when the HAT or SWI SNF and histone chaperone is removed, when this case is happen, pervasive transcription happens. We'll observe the pervasive transcription. Okay. So in this slide, I will explain the underlying mechanism to promote pervasive transcription and also underlying mechanism to prevent pervasive transcription. So usually the chromatin-associated protein, for example, HAT and SWI SNF and also another histone chaperone, they always do the induced histone disassembly. So they can lose the histone, they can lose the chromosome, and also they can do the assembly. Except that they can do the chromosome can be packed. Okay, so assembly doing the transcription elongation. But somehow the chromatin-associated protein was removed. So somehow histone disassembly assembly is stopped. So in this cases, for example, in this cases, this NFR is always open. Okay, in this cases, pervasive transcription can be happened. Okay, so after that, there is another mechanism to prevent pervasive transcription. So when the methylation is happen in histone 3 protein, especially K is lysine, especially 36 location of the lysine is the methylated. So in this cases, the methylation is happens. In this case, we recruit histone deacetylase. After that, histone deacetylase is the decreased histone acetylation. So it means that the chromosome can be very packed. Okay, in these cases, it prevents pervasive transcription. Okay. Okay, today we learned about chapter 1. Chapter 1 is the overview about epigenetics. So we learned about centromere and epigenetics, and also we learned about algorithm and also several things. Finally, we learned about pervasive transcription. Okay, today we will finish the class. Okay, thank you.